Yes.
Yes.
Great.
Yes.
All right. So for the exam 3 review, we'll be going through the blood, heart, blood vessels, and that makes up the cardiovascular unit, the respiratory, and the digestive. So today, or this afternoon session, I am hoping to at least go through blood, heart, and blood vessels, and then we'll see what we can do with the respiratory system. Oops. I'm not sure when the pen's not ready. There's a comment. Let me go back here. See which one is my right. I'm normally able to write on these, but I'm not sure. Here we go. Nope. If anyone uses these things, let me know if you figure out how I'm able to. I think it got updated or something happened and I'm not able to write anymore. Okay, well, we'll do it the old way. Mm. Mm. It's updated and change this on me. Anyone know how to write on PowerPoint? I used to be able to.
If you go to a slideshow and like you present it, will it let you do it like that?
Use slideshow. From slide. Let's try.
And then maybe hit that pin button on like the bottom. Yeah. I don't know if that works right.
I think it does. It's just in the past it didn't make me have to go into there. Right. Because right now you're seeing like 3 boxes, right?
We see like a big one, and then on the right side it shows us the next slide.
Yeah, that's fine. We'll make do with this. Okay. So for your third exam, it's going to include the cardiovascular system, which includes blood, heart, and blood vessels. And then your respiratory, and then your digestive. So if we were to look at the lecture exam, it's going to be the same thing. There's 50 questions, of which the first 40 are multiple choice, and the last 10 are fill in the blanks. Okay. You will have 50 minutes, unless you qualify for extended test taking time. Otherwise, it's 50 minutes. Both the lecture and the lab exam require the respondents lockdown. webcam, and make sure that you did a proper environmental scan. And the reason for the environmental scan is if the respondent's lockdown does flag you, I just watch the environmental scan, and if you show me, you know, to the right, to the left, kind of all over your study space, if there's nothing there, then I just ignore those flags. So lecture exam, 50 questions, 40 multiple choice, 10 fill in the blank. And then for your lab, it is 30 questions, and they are all identification. Make sure if there's two of something that you indicate right or left. Make sure you fill out the complete names. Like when I was grading your quizzes, I noticed that a lot of you forget to put artery or vein right or left. So make sure that you include their complete names, whether they're right or left. If they have 3 parts to the names, make sure you put down all 3 parts. And then you
So with that, sorry, like if we're identifying the right AV valve and the left AV valve, do you want us to spell out atrioventricular if we put like bicuspid, tricuspid?
Great question.
How do you...
However you see it on your list in your lab PDF, I will take those.
Okay.
Yep. And then also I've been noticing that a lot of students use terms that are not listed on the PDF, like odor names. I've been giving you points for them, but make sure that you use the ones that are listed on the PDFs, because they are they match your textbook, as well as they match the most common terms for these structures. Like for example, we used to call Bowman's capsule, now we call it the glomerular capsule. We're moving away from the names of people who discovered or identified them initially to more of logical naming. And that's why I make sure that you are using the terms that are provided for you in the lab PDFs. I think that's it for the general lecture and lab. Any questions on your exam?
I have another question about labs.
Perfect.
So on the video, it says nasal concha, but it just has the A on it on the video. But in my PDF, it has the E on it. Is it just the plural version of it?
That's absolutely right. So either one is accepted.
Okay, thank you.
Other questions? These are all great questions. Other questions? All right. So if you do have any questions along the way, please make sure you ask, because if you have it, most likely someone else has it, but maybe forgot to ask. So that's this is your third exam, so pretty much it's similar to your first and second in terms of the layout and the expectations, and the fact that you can move back and forth. But I don't recommend that. I recommend taking it, using the appropriate time on each question, and moving on, because if you move back and forth too often, that's where the technical difficulties tend to arise. So just kind of go straight through them. See, so that's it for the exam 3. We'll go right into the cardiovascular. So in the cardiovascular, it does include the blood, heart, and blood vessels. So that is 3 chapters. Okay. Starting with blood, make sure you know the functions of blood. So can someone help me identify the functions of blood?
Transportation.
And what does it transport?
Nutrients, gases, and waste products.
Perfect. Let me let the student in really quick. There we go. Go back to my screen. Am I still presenting?
Yeah.
Yes.
Yeah.
Okay. No, it's not letting me do that anymore. So let's go back to current slide. Okay, transport nutrients, waste, and gases. Right. Oh. There you go. What else? What's another function of blood?
Protection.
Okay, how?
The white blood cells fight infections and produce antibodies.
Mm-hmm. What else?
And then
And
clotting.
Yep.
It keeps us from using too much, from losing too much blood in injury.
Yep. And then the antibodies, it's itself. Is it recognized as part of your immune system? Okay. Great.
Mm-hmm.
What's another function?
Regulation of pH.
Perfect. It regulates pH levels. Remind me what's pH levels again? What does that tell you?
The acids and bases.
Right. It tells you how much hydrogen ions are in blood. And the more hydrogen ions you have, the more what your blood is.
Basic. The more hydrogen?
Yep.
Acidic?
Yes. Yep. The more hydrogen ions you have, the more acidic your blood is. Okay? So it regulates pH levels because if it's transporting things, it's essentially transporting the hydrogen ions as well. So it can cause your hydrogen ions to increase in blood, or it can make sure that you are eliminating the hydrogen ions out of your blood through the various mechanisms like your urinary system. Okay? What else does it regulate? It regulates pH levels. It also regulates what? What does it?
Temperature?
Our body temperature.
It can indirectly regulate your body temperature, but that's body temperature is not usually associated with a function of blood.
It also regulates your fluid distribution.
That's it. Yep. Your fluids. So water, since water is the majority of your fluid, right? And water would influence what levels in your body. The more water your body retains, the more what happens? The higher your what? If you don't want to retain the water, you take diuretics. And if you take diuretics, it helps you to remove the water and thus lower your what?
Inflammation?
Not inflammation.
Is it weight?
Indirectly, but that's not what I'm looking for. What's a common ailment in the United States?
Sugar.
Blood sugar is definitely one, but blood pressure is what I'm looking for. It also regulates blood pressure. How is blood pressure related? What's the pressure from in blood?
Pressure against the arteries and veins.
Yep. And what creates the pressure?
Probably the amount of fluid.
Exactly. Yep. So if you have high blood pressure, you're mostly on some type of diuretic medication that helps you to pee it out. Because when you pee it out, you lose water. And if you lose water, it decreases the fluid overall. And if you decrease the fluid, you decrease the pressure. Okay?
Can a healthy body just maintain that on its own?
Absolutely. And it does. Absolutely. And that's where homeostasis comes into play. So if homeostasis recognizes there's too much water in your body, then you urinate more often. You do that naturally, but sometimes it does, some mechanism along the way gets broken, in which case then we use drugs to help us depending on where it's broken to fix it. Okay.
So our water intake influences blood pressure?
Absolutely. Yeah. And that's why if you have high blood pressure, you should have a low sodium diet, because why? Sodium retains water.
Oh, okay.
Yep. All right. So your blood regulates pH level, fluids in general, water particularly, and if it's regulating water, it also regulates blood pressure. Okay. All right. Components. What are the components of blood? So if I were to prick your finger and put your blood into a tube, okay, then I have this cool little machine right here for a centrifuge, and then I put the tubes in there. And what the centrifuge does is that it spins it. And when it spins the tube of blood, it separates the component of blood based on density. Okay? So it's going to separate the blood based on density, so how heavy things are. So I have something here, I have something here, and I have something here. Okay? So let's start with the most abundant. About 55% of your blood is what?
Plasma.
Plasma.
Is plasma. Perfect. And then about 45% of your blood is what?
Red blood cells.
Red blood cell.
Red blood cells. Excellent. Another name for red blood cells is?
Erythrocytes.
Perfect. I love hearing this. Okay. And then less than 1% is what we call the buffy coat. The buffy coat. Now the buffy coat is very small. It's less than 1%. And it's going to include your white blood cells, otherwise your leukocytes, and your platelets. Okay, and it makes sense that it's less than 1%, because in a healthy person, you shouldn't have high white blood cell counts, nor should you have a lot of platelets, right? That makes sense. So the majority of your blood, the lightest is plasma, because plasma is a fluid. Then you have the buffy coat, and then you have the red blood cells. Okay, so that's the components of your blood, right? Now, of the components, we're going to focus in on red blood cells first. So red blood cells, otherwise erythrocytes, you're going to want to make sure you know the function of red blood cells specifically, the structure of a red blood cell, and the life cycle of a red blood cell. All right? So I'm just going to go through it real quickly. It does not mean that that's all you have to know. So whatever details you have, you want to make sure you know them. And in terms of details, I'm referring to what's in your script and the videos, okay? But for the sake of time, I'm just going to go through it, give you a very general overview. If you do have questions, please ask, okay? So functions, now function and structure, this is one of the big things that you should learn from anatomy and physiology, is that function and structure always go hand in hand, okay? Oftentimes, your body is made the way it is so that it can facilitate a function. Okay, so function and structure always go hand in hand. All right. So let's look at the structure of a red blood cell. It looks like a donut. Okay, because I'm a foodie, so everything is in the terms of food for me. Okay. In terms of structure, it's circular and it dips on both sides. So it's discoid and it's biconcave. Bi meaning two, concave meaning it dips inward. It dips here and it dips here. That tells you that the center tends to be hollow. Now, the center of a red blood cell is hollow because normally, what's in the center of a cell?
A nucleus.
Yeah, so that tells you that a mature red blood cell, does it have a nucleus or does it not have a nucleus?
No nucleus.
No nucleus.
No nucleus, that's right. Now, that does make sense because that might be why a red blood cell only lives for about 120 days. Because it has no nucleus. Once it gets damaged, that's it. There's no repair, and it just dies off because it has no nucleus. Why doesn't it have a nucleus? Well, that's the space that gives it space to carry what?
Oxygen.
Yeah. Okay. So no nucleus, which means that that's why it has such a short lifespan of 120 days. And the reason for that is because it needs the space to carry the oxygen, which is why structure follows function. Red blood cells carry oxygen. Okay. It turns out that in addition to carrying oxygen, it also carries what? Not as much, but it also does carry what?
Carbon dioxide.
Yes, ma'am. Okay. So that's the main function of a red blood cell. Now you know there's a process in your body that uses up oxygen, right? What process uses up a lot of oxygen?
ATP.
Yeah. Where is ATP produced?
The mitochondria.
Yeah. So if the function of the red blood cell is to carry the oxygen, you don't want something that uses it up, right? So that's why in addition to not having a nucleus, a red blood cell also doesn't have a mitochondria. No mitochondria. Okay? And the reason for that is because you don't want it to use up the oxygen. Okay? So that does tell you that most of the time red blood cells function under what type of metabolism?
Aerobic.
Aerobic or anaerobic?
Anaerobic.
Very good. Does that make sense though?
Yes.
It's like you would never hire me to deliver Snicker bars. You know those candy, because I love Snickers. By the time I deliver that truck to the grocery store, I would have eaten all the Snickers. No point, right? Same thing. So that's why it wouldn't have mitochondria, because the purpose of a red blood cell is to transfer the oxygen, not to use it. Okay? All right, so no nucleus, no mitochondria, it's biconcave, it's discoid. Okay, so we're good so far. Next. Oh, I'm sorry. Questions before I move on? Okay. So let's focus on the life cycle. Now, as we're doing the life cycle, I actually also want you to walk me through hypoxemia. Because why not, right? All of you did, not all of you, but some of you chose to do hypoxemia and you did such a great job. Walk me through it. What's hypoxemia? Hypo means what? High or low?
Low. So it's like low oxygen in your blood, right?
That is exactly it. Low oxygen in blood. Okay? Which is bad, right? Because you need oxygen to make ATP. So that's why oxygen is important. Okay. A lot of people think it's the breathing that it's important, but really it's not. It's the fact that you need the oxygen for your ATP. You're not like a flashlight where if the battery runs out, you can just put new batteries in and then it starts again. No, once your battery runs out, you cannot bring it back. So it's not necessarily the inhalation expiration that's important. It's the oxygen. Okay. All right. So hypoxemia is low oxygen levels in blood. So what's going to happen is that homeostasis is going to sense that you have low oxygen levels in blood. Okay, what's it going to do?
Hypoventilation.
No. Internally, what is it going to do?
Is it going to like cycle the blood faster?
No.
Is it like a higher amount of carbon than O2 within the blood?
You're thinking like a healthcare professional. Think like an anatomist. Okay, so all of you, what you had said are stress results. So you're right. When someone is physically having the conditions of low oxygen levels, they're going to hyperventilate as a result of it, right? Because they're trying to bring in more oxygen. And if they hyperventilate, they're trying to bring in more oxygen, but what actually happens is that they excrete more carbon dioxide. So psychologically, that does happen. Okay, but that's more of like a healthcare, which is great because all of you are going into healthcare. But think in terms of anatomy. So internally, what's happening? So if your brain senses low oxygen levels, it's automatically going to do what? It's going to release what?
Does it have to do something with iron?
Not iron. But you're getting closer because now you're going internal.
Hemoglobin?
Closer. You want more hemoglobin because it figures if you have more hemoglobin, it's going to be able to carry more of the oxygen. So how do you get more hemoglobin?
Homopoiesis?
Yes. Okay. What triggers hematopoiesis?
Low oxygen.
How does low oxygen trigger hematopoiesis? I'm looking for something. If you ever remember Lance Armstrong, he got in trouble for
doping.
Exactly. And
Yeah.
what did he dope with?
I can't remember what it was.
It's a chemical.
I just finished watching the Tour de France yesterday.
Yes, there you go.
I've been a cycler since I was a teenager.
Yep.
Gosh.
Some sort of steroid.
It starts with
Yeah.
an E. Your body naturally makes it, but athletes sometimes they put more into their body than they naturally make. Erythropoietin. Do you remember that term, erythropoietin?
That's right.
Okay. Erythropoietin naturally is made by the kidney and the liver.
Yes.
Okay. So hypoxemia is going to trigger the release of erythropoietin. We're going to just put EPO for short. Okay. Erythropoietin from the kidneys and the liver. The erythropoietin goes where to make more red blood cells?
To the bone marrow?
Yes. We'll say the bone, right? Because not the bone marrow doesn't necessarily always make red blood cells, right? Right? Yes?
Right.
Yes.
Okay.
Right.
Good. Where else can you make red blood cells? You have bone marrow in the medullary cavity, but you also have some here, right?
This relates to the bones that we talked about in the other chapters, right?
Absolutely. They're all connected. That's why anatomy and physiology is like my favorite thing to teach because everything is connected.
Mm-hmm.
Yep. Okay. So the erythropoietin that's released due to low oxygen levels target the bone marrow or just the bone in general, and that's going to increase red blood cell production. Okay. So if you increase red blood cell production, of course, inside the red blood cells, what's the functional unit of red blood cells?
Hemoglobin?
Yeah, exactly. Okay. So that's one way in which red blood cells are produced. Okay, is through this process as an example. Or it could just be in general, a red blood cell being produced. Okay. But essentially this happens all the time in your body. New red blood cells are constantly being made. So can you afford to donate blood?
Yes.
Absolutely. Yep, because your body will just release more EPO. EPO will cause the red blood cells to be produced. More red blood cells produced. It only lives for 120 days anyway, so this cycle continues onward. Okay? So once the red blood cell is produced, it's got lots of hemoglobin. It's going to survive for how long?
120 days.
Perfect. 120 days. And it's going to get damaged. Why does it get damaged?
So the pressure of the where it passes, I saw a video on this. They pass through
Yeah.
like blood vessel and they get damaged and they don't have a nucleus to regenerate. So it just die off.
Excellent. Excellent. You got it. You give the rest of you lecture your peers because you got it. No, I'm serious. You did a really good job. Okay. So what she said. So as it's moving through the membranes of the tissues, it has to squeeze through the little gap openings, and then the membrane just gets damaged. The membrane of the red blood cell gets damaged. It doesn't get repaired because it doesn't have a nucleus. Okay. So that's why in about 120 days, it starts to break down. In your body, once it breaks down, it's going to be collected by this little organ right here or this larger organ right here. So it's either the liver or the spleen. The liver and the spleen have these fibers right here, this mesh that collect red blood cells that have been damaged. Okay. By the way, a nickname of the spleen is called the graveyard of red blood cells. Graveyard of red blood cells. Because really that's its main function. Okay, is that it collects all the red blood cells that get damaged and it breaks it down. When it breaks down the red blood cells, it's going to break it down into its functional units, which are hemoglobin. Okay, and hemoglobin gets broken down further. And this is one of the questions that was requested, so I'll spend a little bit more detail on this one. So hemoglobin gets broken down into the heme groups and the globin chains. The globin chains essentially are just amino acids. They're going to be broken down even further into amino acids. And the amino acids are reused to form, other globin chains or other protein because they're amino acids. Okay? The heme group is what's interesting. The heme group, there's 4 heme groups. There's 4 hemes in general that looks like this. 1, 2, 3, 4. Okay, so the heme groups, there's 4 of them. Each one of them have an element attached to it, which is what allows it to carry oxygen. What element is essential for the hemes to carry oxygen?
Iron?
Yeah,
Iron.
that's right. Okay, heme groups will not carry oxygen unless it has the iron. Think of it this way. In the past, cars used to be made of metal, right? And over time, that metal rusts. Why does it rust? Because it has
It's
a high.
old.
It's old, absolutely. It's old, but also because metal has a high affinity for oxygen. Okay, so it oxidizes the metal. All right, so the iron, the reason why iron is on the heme groups is because iron has a high affinity for oxygen. So the oxygen is going to bind to the iron. Okay, so one molecule of hemoglobin can carry how many molecules of oxygen?
1.
4.
4 is the correct answer. One molecule of hemoglobin has 4 heme groups. Each heme group has an iron. So technically, 4 oxygen can bind to the 4 irons, 1-to-1 ratio. Okay, so one hemoglobin can bind to 4 molecules of oxygen. Okay, all right, so that's what the heme groups looks like. We mentioned earlier, though, that hemoglobin also carries carbon dioxide. Where does this happen? It's
In the
not on the
gas
heme group.
exchanges?
Say that one more time.
In the gas exchanges?
Where does it? What structure binds to the carbon dioxide? What's remaining?
The globin chain.
Yes, ma'am. So the globin chains are responsible for binding to the carbon dioxide, where the heme groups are responsible for binding to the oxygen. But it should make sense at this point. Why? Because the oxygen binds to iron and iron binds to the hemes. Okay? So technically, the heme group is made up of the iron and these hemes, which are pigments. Okay? Pigments, so coloration. Okay? So when you break the globin chains down, you break them down into the amino acids, which are reused. When I break the heme groups down, I break it down into two parts. What are the two parts I break it down into? The heme pigments and the iron. Okay? The heme pigments are going to continue onward towards the liver and become things like bilirubin, and then bile. So eventually it's going to become bile because it's going to head to the liver. Okay? And then as bile, it's going to excrete out through your feces. The iron, you're going to want to either reuse it or you're going to lose it through trauma, through blood loss. Okay? What gender experiences blood loss every month?
Females.
Yes, that's right. And that's why oftentimes females more than males have anemia because they lose the iron through their menstruation. Okay, I'm sorry, I have to let the student in. Give me 1 second. Welcome. Okay, so the iron is lost. We want to reuse it, but unfortunately, it is lost through blood. We lose it through blood loss. Okay, and that's why women tend to be more anemic than men because of the monthly periods. Okay.
Is that why people take iron supplements to assist with like the hemoglobin groups?
You got it. Good connection. Yep. Yeah. Okay. And that you'll notice that I try to connect it to conditions of the body to help make sense to make the science make sense. So as you're studying, try to see if you can make connections to something that helps solidify that information. So it's not so much as memorizing all the details, but then it's going like, oh, that makes sense. Okay. All right. All right. So that was red blood cells. We're gonna move on and talk about plasma. So in plasma, make sure you know the functions, the components, and the plasma proteins. Okay. I'm going to leave functions and components, but I do want to focus on plasma proteins because that's what sometimes students have the most trouble with. There's a lot of plasma proteins, but I only require you to know 3 of them. What are the 3 plasma proteins that I require you to know?
The fibrogen, the albumin, and the globulin.
Yes, ma'am. I love how you just listed that like it was nothing. Fibrinogen, albumin, and all of you have to be able to do what she just did. And globulin. Okay? So fibrinogen is an inactive form of fibrin. How do you know it's inactive?
Because it's got the gin on the end of the word.
That's right. The ogen. Yep. Anytime you see the word ogen, it means it's inactive. Pepsinogen, fibrinogen, all inactive, which makes sense because you don't want to have clots happening normally in your body, right? You only want it to happen when you need it to happen. So in your blood, this plasma protein is inactive unless you have a cut. And then there are proteins on the outside of your blood plasma that enter and activate the fibrinogen into fibrin because there's a cut. So now you need it, right? But normally you don't want it to be active. I think someone joined the group. Sorry, let me put them in. Okay. Okay, yeah. So fibrin, what does fibrin do?
Creates like a mesh on your skin whenever you cut yourself.
Yeah, it's a clotting factor. Okay, great. How about albumin? What does albumin do? So my dad used to undergo, oh, sorry, go ahead. What were you going to say? Hmm? Don't worry, this is the best time to be right or wrong, right? It's better, and actually studies show that if you're wrong when we're doing our study sessions, that you're more likely to get it right when you take the exam because you remember it more. Okay, so my dad had to go to dialysis all the time because he had ascites. So ascites is fluid in the stomach. Okay, so what they did is that in paracentesis, they would take the fluid out, and then afterwards they would give him albumin. And the reason for that is because the albumin helps to pull the water back into the blood vessels so that he doesn't get dehydrated. Okay, so albumin is often a chemical that you can give to patients that undergo paracentesis or any other types of things where they're afraid that their blood would be too dehydrated. Okay, and the reason for that is because albumin impacts or is a factor in viscosity. I lost my pen. I cannot write my pen anymore. In viscosity and osmolarity. So I'm writing with my mouse now, and it's not looking very pretty. Viscosity and osmolarity. In other words, it controls how thick blood is through the movement of water. Okay, so albumin controls viscosity and osmolarity, in other words, how thick water is. Okay, then the last one is globulin. Now, do make a note that the faculty that presented on globulins, they kind of made it too general because they talked about globulin overall when technically there's 3 types of globulins. There's alpha globulin and beta globulins. Alpha and beta globulins are the ones that are responsible for transportation. So they bind to things in plasma and they help move it along. But you also have a third one, okay, which is gamma globulins. Now, gamma globulins are your antibodies. So it's these specifically that function in immunity. Okay? So make sure you distinguish the globulin types because the faculty just said globulins function in transportation and immunity, which technically is true, but you have to know by the types. So it's alpha and beta globulins that do the transportation, where it's gamma globulins that are antibodies that function in immunity. Okay? And just the gamma can do that, where just the alpha and beta do the transportation. All right? So those are the 3 plasma proteins that you should know. In terms of white blood cells or leukocytes, there's a mnemonic that helps you remember. Never let monkeys eat bananas. Okay? I really like this mnemonic, even though it's kind of cruel to monkeys, but it allows you to know the components of white blood cells from the most abundant to the least abundant. Okay? N being for what?
Nasophils
Neutrophils.
or neutrophils.
Okay, yeah. Neutrophils. Okay? And neutrophils would indicate what? So if you have elevated numbers of neutrophils, what does that indicate?
You're fighting an infection.
What kind of infection?
Bacteria.
Bacterial infection.
Bacterial.
That's right. And you should know that, right? So you should know the specifics of it. So neutrophils does fight infection, but specifically it's elevated when you have a bacterial infection. And that's good to know. And that's why they do your blood work. If they find that you have elevated neutrophils, they may not necessarily wait until they figure out what exact bacteria is causing your infection and just throw some broad spectrum antibiotics at you. Okay? Because you may not have the time to figure it out. Because it takes a long time to figure out what specific bacteria. Okay? Then you have L. L stands for what?
Lymphocytes.
Yes, perfect. Lymphocytes. Lymphocytes are like the main, the main function of your white blood cells. Lymphocytes will become T lymphocytes and B lymphocytes. Okay, so they differentiate. They become various different types of cells that have various different types of function. Okay, so the lymphocytes, not the H, lymphocytes become T cells and B cells, but essentially the lymphocytes are your main immunity. It's going to protect you against cancers. It's going to protect you against viruses. It's going to protect you by producing antibodies. So that's why your lymphocytes are kind of like your main immunity. Okay. Then you have M. M stands for what?
Monocytes.
Yeah. Mm-hmm. Monocytes are also important. What are they good for?
Fighting off viral infections.
Yeah, specifically viruses. Okay, so you would go through and you would do that for E, eosinophils, B, for basophils, and identify what they do. Okay, now of these, L and M are agranulocytes, meaning these cells don't have granules in them. A means without, granulocyte meaning granules. Okay, so make sure you describe the white blood cells and its function. The heart, I think a lot of you have a strong understanding of the heart. With the 10 minutes left, I want to go through, let me see what I wrote down is for requests. So heme groups I covered. Venous, okay, let's do, venous return would be part of, venous return and ventilation. Let's wait for the evening when we do digestive, okay? So I'm going to finish off, I'm not going to do heart structure and blood flow because that takes a lot of time. And plus, I think most of you have a strong grasp of that.
I have a quick question.
Yeah.
It's over the cytoplasmic granules.
Yeah.
So how the organulocytes do not have cytoplasmic granules, but the granulocytes do.
Yes.
What exactly are the cytoplasmic granules?
Yeah. So they're protein structures. So here's the nucleus. Let's say this is a neutrophil. Neutrophils actually have multiglobule nucleus like this. Okay. So there are proteins inside that makes it look like freckled,
Mm-hmm.
That's the granules. Now, each cell has different types of granules. Like, for example, basophils would have things like histamines and heparin, okay? Where neutrophils have their own granules, and these granules help to actually destroy whatever they're fighting against. So this one would be bacteria, for example, okay? Where lymphocytes and monocytes work by swallowing up the viruses, or they use different mechanisms to destroy, where granulocytes use their granules to actually destroy the infection.
Oh, okay. That makes sense. Thank you.
Yeah. Okay, so with the last few minutes, I want to talk about the blood vessels. I noticed that a lot of you did the video on this, and your videos are great beginnings, but you need to be able to go in deeper into the details. So what I was looking for, and some of you did this in your videos, and some of them were a little bit more general, but this is what I was looking for. You want to know the function, right? A few of you said arteries are always oxygenated and veins are always deoxygenated, which is not true. Mostly true, but not always true. So you have to be really careful about that, okay? So in terms of definition, you cannot say that all arteries carry oxygenated blood. 99% do, but not all. And that's why the definition of an artery is more specifically that it carries blood away from the heart, A in a way, A with artery, okay? And veins, instead of saying that they carry deoxygenated blood, you should say that they carry blood up towards the heart, okay? Now, the reason why you can't say that arteries carry oxygenated blood and veins carry deoxygenated blood is because in the heart itself, you do have arteries and veins that carry the opposite. So in this case, here's an artery. It's carrying blood away from the heart to the lungs. And in this case, these arteries right here are going to be deoxygenated. And it turns out these are the only ones that are deoxygenated, but because of that one artery, you can't say that arteries carry oxygenated blood, even though 99% of the time they do. Likewise, in the heart, you have veins coming from the lungs that now carry oxygenated blood back to the heart. And because of that, you cannot say veins carry deoxygenated blood because there is one set that does not. Does that make sense?
Yes.
Okay. You also need to know the details of the layers, which a lot of you skipped on. Okay, so a lot of you forgot to talk about the layers. So veins and arteries have 3 basic layers. They have the internal layer, they have the media, and they have the externa. But there are differences within these 3 layers between arteries and veins. And it's those differences that give it its unique characteristics. Because remember, structure follows function. Okay, so you need to know why, how are they different? And that's what I was really getting at, is for you to compare and contrast. Yes, we know that both of them have the 3 layers, but how are they different? How are they similar? Okay, so some of the major differences is that veins have valves. Why? Because at the time that blood is moving through the veins, there's very little pressure. And because of that, it has to have things that help move the blood in one direction, and that's why it has valves. In addition to that, the tunica media is going to be thinner, okay? Where in arteries, the tunica media is a lot thicker, okay? And the reason why it's thicker is because it does have a lot of pressure, so it has to be able to withstand the high pressure, otherwise the blood vessel would rupture, okay? And then you also want to be able to, okay, so in this view, you can't see the bottom, but the bottom has capillaries, okay? So that's it for these 3 chapters on blood, heart, unfortunately we had to skip, and then blood vessel. So in the respiratory system, a couple of things that you'll want to know will include the function structure of the respiratory system. The best way to do that is through the cognitive map that I included in the worksheet that I sent out to everyone. Okay, so with the cognitive maps, it allows you to walk through kind of all the steps from when you breathe in air to when it leaves your body. So just kind of a quick review. If we were to follow a molecule of oxygen from the outside into your body, what's the first thing it will go through?
Your nose.
Keep going.
into the pharynx, larynx, trachea, bronchial tree, into your lungs.
Larynx, trachea. And a good mnemonic to remember that is please let's talk. So please let's talk. Nose, pharynx, larynx, trachea. From the trachea, it goes to the bronchial trees. And then you have the primary, secondary, tertiary, or the main lobar segmental. And then from here, it goes to the bronchioles. And then it makes its way through the different types of bronchioles until it finally gets to the lung, and more specifically, the alveoli. Okay. And then when you breathe out, it would go the opposite direction. Okay. All right. So you would essentially want this list and then be more detailed in terms of what accessory structures you would find along there and what tissue lining. And same thing, the tissue lining seems really hard, but once you kind of get the hang of it, you'll find that the tissue lining actually matches with its function. So for example, along the pharynx, if it's just air, it would be pseudostratified columnar epithelium. If it allows for both air and food and beverage and food in general, it's going to be lined differently. Okay, so in that case, it would be lined with what?
Stratified squamous epithelium.
Perfect. Exactly. So it should be less of a memorization and it just should make more sense. Okay. All right. So what I'm going to focus on is on ventilation, but also make sure you understand about gas exchange. So with gas exchange, you do need to account for two. You need to account for the pulmonary gas exchange, as well as the systemic gas exchange. Okay. So go ahead.
Sorry, is the pulmonary gas exchange the same thing as the alveolar gas exchange?
Yes.
Okay.
Yep. So the pulmonary essentially is between the blood vessels and lungs. Anytime you see the term pulmonary, it has to do with the lungs. Where the systemic one is essentially the blood vessels and the body. Okay. All right. So let's focus on ventilation. So in ventilation, actually a lot of you did really excellent videos on this. So I highly recommend your colleagues' videos because sometimes they explain things a way that clicks better than how I would do it. But essentially when I talk about ventilation, which is breathing, okay, there are a couple of things you need to know before we go into the actual mechanisms of ventilation, and that is the following rules or laws, okay, which include what? You've got Boyle's law, and what's Boyle's law?
Something that has to do with the pressure gradient.
That's right.
Mm-hmm.
Pressure and what?
Pressure in the environment and pressure inside.
Yep. Pressure and what?
Pressure.
Volume.
Volume. So pressure, Boyle's law states that pressure and volume are related in what way?
Aren't they opposites?
That's right. Inversely proportional. So opposites is a good way of saying it actually. When one goes up, the other one goes down. And you can think of pressure as force. So it's the force that's put onto structures, okay? A good way to think about this, the analogy that I give about this is a can of soda. So in a can of soda, I have, what's normally in a can of soda? 12 ounces? 12 fluid ounces, okay? Now, if I take a gallon of milk and I poured that 12 ounce in here, okay? So I changed the volume, right? So what I did is that I increased the volume. So when I increased the volume, what happened to the pressure?
You decrease the pressure.
Exactly. So in that can of soda, the volume is low, but the pressure is what?
High.
Hi.
Exactly. And that's why if you shake that can of soda and you open it, it spurts at people, right? Because the pressure is so high. But if I use that same amount of fluid and I put into a larger space, a larger volume, then the pressure goes down. Why is that? Well, pressure is formed by force. So the molecules in here, what happens is that when the molecules bump into each other, that creates the pressure, the force. So if I increase the space, then the likelihood of these molecules bumping into each other is less. Okay, so that's why when volume goes up, pressure goes down. Just like imagine you're in a room. Okay, so if I put you in a small room, a 12 by 12 room, and I told you to walk around, and there's like 5 of you, the likelihood of you walking into each other is much higher than if I doubled the space. And imagine whenever you walk into each other, that creates pressure. And that's why volume is always inversely proportional to pressure. Volume goes up, pressure goes down. Make sense?
So that's exactly what happens in inspiration and expiration, correct?
Partially.
Okay.
So partially, that's one of the rules. The other rule is diffusion. This one you've learned a long time ago, right? Okay. So in diffusion, someone remind me what's diffusion again?
You don't need
Movement from high to
CP.
low concentration.
Yep. High to low concentration gradients. Okay. Is it until the other side becomes high or just until equilibrium is reached?
Isn't it till it's in equilibrium?
That's right. Okay. All right. So between these two rules, in addition to that, I also have key muscles. What are the key muscles in ventilation?
The diaphragm and the intercostal muscles.
That's right. The diaphragm and the intercostal muscles.
The thoracic cavity wouldn't go in there too?
The cavity is a space, right? So whenever you use the term cavity, it's a space. So that space is a factor, but it's a factor in Boyle's law,
Okay.
right? So it's not a muscle. Okay, so the muscles that create that space is the diaphragm and the intercostal muscles. Okay, all right. So because we live at sea level, the atmospheric pressure is going to be pretty consistent. What's the atmospheric pressure? Was the atmospheric pressure? Okay, 760 millimercuries. Okay, so it stays pretty consistent. Okay. So a person's diaphragm is naturally curved, like so. Okay. Now, when I contract the diaphragm, am I going to cause the diaphragm to get shorter or longer?
Longer?
Longer. Oh, no. When I contract the diaphragm,
It's getting shorter.
it's getting short. Contraction causes muscles to get shortened. Okay. So what's going to happen is that diaphragm that's naturally slightly curved upwards is going to pull downwards. It's going to be right about here. Okay, so here's the change in volume. So when the diaphragm contracts, as well as the intercostal muscles, okay, what does that change? What change does that do?
There's more space.
That's right. And what is
volume?
There you go. So the volume increased. And when the volume increases, what does that result in?
Pressure decreases.
That's right. So the pressure of the lungs, okay, it's going to go from 760, where it's neutral, so it's going to go all the way down to 7 what? 756. Okay, so that's the first rule that we just applied, is Boyle's law. Volume goes up, pressure goes down. Now I'm going to use the diffusion. Now the pressure in the atmosphere is 760, where the pressure in the lung is 756, which is higher.
Outside.
That's right. So then, according to diffusion, which direction will air move?
From the lungs to like outside the atmosphere. Sorry, from the atmosphere to the lungs, high to low.
Right? There you go, from high to low. Okay? So that's inspiration. A lot of people think that inspiration occurs when you breathe in and that causes your lung to expand. That's not what happens. Your lung expands first, which then changes the pressure, and then the air is sucked in. That's what inspiration is. Okay. A lot of us reverse it. We think that in order for inspiration to occur, your lung expands. I'm sorry. We think that in order for inspiration to occur, the air moves in and that causes your lung to expand. Your lung expands first, and that's what causes the air to move in. Okay? So that's inspiration. Now in expiration, the diaphragm, instead of contracting, the diaphragm is going to do what?
Go back to the
What's the diaphragm going to do?
Relax.
It's going to relax. And when it relaxes, does it get shorter or longer?
Longer.
Longer.
That's right. So now it's going to get longer and it's going to push upwards even more. So what change did it do?
It changes the volume in your lungs.
To what? Does the volume increase or decrease?
Decreases.
Decrease.
And when the volume decreases, what happens to the pressure?
Increases.
increases.
So the pressure of the lung is going to go up to about 763 millimercuries. So now, compared to the atmospheric pressure and the lung pressure, what's going to happen in diffusion?
It's going to move high to low. So since the lungs are higher, it's going to move out.
And that's expiration. So that's ventilation. Okay, so you know there's going to be test questions on this. So the test question could be, in inspiration, does volume increase or decrease? Does pressure increase or decrease? How would you answer that? In
The
inspiration.
volume's going to increase, so pressure will decrease.
Perfect. That's exactly it. Okay, and that actually is one of the test questions. Okay, I'm not sure if you're going to get that question because again, the exam is randomized. You know, you just get random questions, but yep, that has been a test question in the past. Okay. All right. Any questions on ventilation? Hopefully that clears it up a little bit. I feel like a lot of you got the understanding of it by watching your videos anyways. Okay. All right. right. So I'm going to leave the last 40 minutes for digestive. So is there any requests for digestive? Anything specific you want me to cover?
Yes, I had a question in chemical digestion.
Mm-hmm.
Like when they turn into their building blocks and absorption and all that. Like there's a little graph at the end of our chapter.
Mm-hmm.
And
Yep.
it's really confusing what does what and where and all that.
Okay. So you want to focus on chemical digestion. We can absolutely do that. Yep. Okay. So for the digestive system, you want to be able to describe the function. Oops. So what are the functions of the digestive system?
Ingestion, digestion,
absorption, compaction, and defecation.
defecation.
Perfect. Let's start with ingestion. What's ingestion?
Eating.
Yeah, that's my favorite part of living, eating. And then you have digestion. And what's digestion?
The
Breakdown.
breakdown of food.
And how does that occur?
By
Either
enzymes?
mechanically or chemically.
There you go. That's the answer I was looking for. There's two forms of digestion. So that means that on the test, you have to be able to distinguish between those two. So there's mechanical digestion as well as chemical digestion. Mechanical is just the physical breakdown. Can you give me some examples of mechanical digestion?
Like
Chewing.
chewing, turning, grinding.
Peristalsis.
So mastication or chewing happens where?
In your mouth with your teeth.
Yep. The churning occurs where?
The stomach.
The stomach.
Yep, exactly. Okay. And then chemical happens throughout. And usually that chemical is in the form of what? Someone said it.
Enzymes.
Enzymes.
Yep. And how do you know it's enzyme? It always ends in what?
The
ASE.
ACE.
Yep. ASE. ACE. So enzymes always end in ACE. Well, not always, but most of the time ends in ACE. Okay. All right. So when we're talking about chemical breakdown, you need to know the name of the chemical. You need to know what it breaks down. And you need to know where it is produced / secreted. Okay. So a couple different layers to track. Okay. So that's digestion. Then after digestion, you have absorption. How do you define absorption?
Like the uptake of nutrients into
Yes,
the rest of your body.
uptake of nutrients, lipids, water, vitamins, etc. And when we say into the body, technically isn't it already in the body?
No, until we get into the small intestine. No.
It's like into the bloodstream.
There you go. So technically, your whole digestive tract is just allowing food to move through your body, but it's not in your body until it is absorbed into either the bloodstream or the lacteals. What goes into the bloodstream?
Carbs and proteins.
Yeah, carbs, amino acids, nutrients, certain vitamins, water. What goes into lacteals?
Fats.
Yes. Okay. All right, so that's absorption. then at whatever's left does what?
It gets compacted for defecation.
That's right. Compaction and defecation. Great. So that's the function of the digestive system. Okay. So same thing. Go through the digestive tract. You can do the same thing we did for the respiratory tract. You can number it. So if you were to follow an Almond Joy candy bar, how would it move through your digestive tract? So from the mouth to the pharynx to the esophagus, so on, so on. Okay. So use the cognitive map. Okay. And as you're using the cognitive map, make sure you also know the lining. Okay. Why am I making you know the lining? Because the lining correlates with the function. Okay. So you learned about the alimentary tract, about how it has the mucosa, the submucosa, the muscularis, and the serosa. The entire digestive tract, from the mouth to the anus, all have those 4 layers. But those 4 layers change depending on what area you're looking at. So the stomach in the muscularis layer, instead of having 2 layers of muscle, it's going to have how many?
3.
3.
Exactly. Because the stomach has to do the churning. Okay.
Mhm.
And in your small intestine, okay, you have the villi. And why? Why do you have the villi along the mucosal lining?
For absorption.
Exactly. It slows down the movement of food, increases surface area so that food can be more easily absorbed. Do you find villi anywhere else? You don't. Okay. So there's unique structures to each portions, and that's what defines that portion as an organ, as a different organ. Instead of just like saying that the whole thing is digestive tract, we have the esophagus here, we have the stomach here, we have the small intestine here. Why? Because along that digestive tract, the alimentary canal, the tissues that make up that tract, it changes. And the changes indicate its function. And that's why you have to know the lining. Okay? All right. So go through the cognitive map, list out the pathway of food, and explain how the lining correlates with the function. Okay, because it does. All right. So that's this next bullet point right here. So describe the function of the mouth, the sensory organs, the tongue, the lingual papilla, the lingual frenulum, the palate, the uvula, the tooth, the teeth, and the structures of the teeth. You've got the root, you've got the crown, you've got the enamel, the tissues that form them, what's the living tissue of the teeth, what are the salivary glands, what do they secrete, and don't just write down saliva, because you have to know the components of saliva as well. Know the pharynx, know the esophagus, the stomach, the small intestine, the large intestine, until finally it exits the body. Okay, so take a look at these questions and be able to answer them in general. Okay. Then lastly, which is what we're going to focus on in this last part right here, will be the chemical digestion. Okay. So I always like to use examples instead of like going through what they are. And a good way to do this is just to pick a food. Now I picked Almond Joy because it's like one of the best candy bars. You can argue with me, but it's one of the best candy bars. And the reason why I picked that is because it has all 3 components. So the chocolate represents the carbohydrates. The almond represents what?
The protein?
Yes, ma'am. And the coconut represents what? What?
Fat.
Fats.
Yeah. Did you see that? One candy bar with all the food qualities. That's what I tell my family. That's why we all have to eat Almond Joys. Okay. Anyways, so follow it through and we'll go through it together and we're going to identify what is broken down by what. Okay. So we'll start in the mouth. You have the tongue, the teeth, the salivary glands. So In terms of function, it allows for ingestion and digestion. Whenever we talk about digestion, you have to tell me, is it physical, is it chemical, is it both? In here, it's both. So it's physical digestion through mastication, so chewing. It's chemical digestion because your mouth secretes saliva, and saliva includes the chemical that it includes that allows for chemical digestion is salivary amylase. ACE enzyme, salivary because that's where it's produced. AMA, that's what it breaks down. Amylase, it means carbohydrates. Okay, so salivary amylase in saliva is what breaks down the carbohydrates or the chocolates. Okay, so have you ever wondered why there's a difference between when you eat rice only or when you eat meats only as to which one causes you to feel full longer? This is why. Which one do you feel fuller with longer? When you eat rice or when you eat meat?
Protein, meat.
Protein.
Right, because protein is not digested until it gets all the way down to your stomach, where carbohydrates are digested right away in your mouth. So you absorb it faster, so you feel full shorter. Okay, meats get digested a lot. It's harder to digest, so it stays in your digestive tract for a longer time, so you feel full longer. Okay, all right. So in the mouth, you've got salivary amylase that breaks down the carbohydrates. Now, it turns out that saliva has more than just salivary amylase. Okay, so what are some of the components of saliva? So we know salivary amylase is one. What else? You've got water. What else?
Water and mucus.
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 This. What else? You've got IgA, which is an antibody. Okay, so that's part of your immune. So right away, we know over time that whatever we eat, it's got bacteria and it's got some microbes in it. So we have some immune functions right away. Okay, what else do you find in here? What did I just write?
Lipase.
Yeah. What do you know about it?
I mean, it's an enzyme since it ends in SC.
Yep. What does it break down?
Fat.
Fats. It breaks down lipids, right? So lipase breaks down lipids. Now, it is secreted in the mouth, but it is not active until when?
Is it in the small intestine?
Not the small intestine, a little bit earlier.
The
The
stomach.
stomach?
Yes. Okay, so it's got to have an acidic environment to activate it, essentially. Okay, so that's why even though salivary lipase is produced and secreted in the mouth, it doesn't do anything. The only thing that's active is the salivary amylase. Okay, so at this point, whatever you've chewed, what is the food called at this point?
Bolus.
Chime.
Bolus is the correct answer. Yep. So the bolus is going to move from the mouth down the pharynx, and specifically the oral pharynx and the laryngeal pharynx. Okay. From there, it's going to move to your esophagus. Make sure you know the change in lining of the esophagus. It's going to start from skeletal and become smooth, okay, muscle-wise. And then from the esophagus, it's going to go into the stomach. So the pharynx and the esophagus is just pathways. So there's no ingestion, no digestion, no absorption, nothing. It's just going to move along. How is it going to move along?
Through the waves, peristalsis, something like that.
Yes, peristalsis. Okay. So it's going to move from the esophagus to the stomach. Okay, so the stomach looks something like this. Looks like a J shape. So this opening right here is called the cardiac. The dome of the stomach is called the fundic or the fundus. Then you've got the body, and then you have the pylori.
Pylori.
Yep. Okay. So in terms of function, digestion happens here. And whenever we talk about digestion, you have to tell me whether it's physical, chemical, or both. And it's going to be both. So physical, through the contractions of the rugae or the smooth muscle. Okay. Where do you find the smooth muscle? In the stomach? What layer?
obliques.
Muscularis, right?
Yes, that's correct. Muscularis. And it has 3, right? What are the 3?
Longitudinal, circular, and that's where the obliques come in.
Yes. Okay. So you've got the longitudinal, the circular, and the obliques. And all in the muscularis. And they are all smooth muscle. Okay. So the contractions of them leads to the churning or the physical breakdown. You also have chemical breakdown. You've got pepsinogen that breaks down into pepsin, and pepsin breaks down proteins. It's going to break proteins down to polypeptides. Okay. Polypeptides. So it's going to essentially break down the almonds. Okay. How does it convert from pepsinogen into pepsin? Remember, whenever you see the term ogen, it means what?
Not active.
That's right, inactive, right. So the hydrochloric acid has several functions. One, it's going to help break things down, and it also helps against bacteria, and it actually converts pepsinogen into pepsin. It will also activate the salivary lipase. Okay. Then you also have gastric lipase. So between the salivary lipase and the gastric lipase, there is minor digestion of lipids occurring here. Okay. Questions?
So in the gastric lipase, there's just lipase is fat, right?
Yep.
So there's like minimal digestion of lipids. What does that mean? Like they don't fully digest it?
Right. They just break it down to smaller lipids.
Okay. And then the rest happens in the small intestine.
That's right.
Okay.
Because lipids are like adipocytes, tons and tons and tons of adipocytes.
Mm-hmm.
Clumps of it.
Mm-hmm.
So essentially it just breaks it down to a smaller clump.
Okay.
Yeah.
All right. Thank you.
Yeah. Okay. Then we have the small intestine. The small intestine is going to include the duodenum, jejunum, and ileum. Okay. So a good mnemonic to remember this is don't jump in. So don't jump into the small intestine. Duodenum, jejunum, and ileum. Now, as we're going through this, you can be really detailed because there's a lot of information in the small intestine. Okay. You know, normally if I were to lecture this, I could spend an hour on just the small intestine because you have to go through the lining and then the unique structures within the lining, like the villi, the circular folds, And then you can go through the cells that form them and so on and what the cells produce. So these are things that you will want to make sure you review. But for the sake of just chemical digestion, I'm just going to focus on chemical digestion. Okay? So in the small intestine, interestingly enough, it actually doesn't make a lot of things. What does make a lot of things and dumps into the small intestine is the pancreas, the liver, and the gallbladder. Okay? So let me add one here. So here's the stomach. Kind of like goes around and forms this large tube, so on. Okay. Now, I wouldn't be able to tell you where the jejunum and where the ileum is. You'd actually have to take it apart and measure it. Okay. But I can tell you that this portion right here, is going to be the what?
The jejunum.
Yep.
Duodenum?
Yep, the duodenum or the duodenum. You can pronounce it any way you want because I've heard it be pronounced both. All I know that is the duodenum. And I like saying duodenum because it helps me write it out. Duodenum. Okay, or you can say duodenum. So whatever helps you, for me, it's duodenum helps me to spell it out. So that's how I pronounce it. So the duodenum or the duodenum, that's the only place I know. With the others, I know it's somewhere in there, but I'd actually have to take it apart and measure it to know what exactly is the jejunum and what's the ileum. Okay? All right. Now, it turns out that I have a couple things being dumped into here. The one through here, through this organ, and also through here. Okay. So I've got the pancreas over here, and I have the liver here. And then I have the gallbladder here. Right? So what does the liver do?
It produces bile.
Mm-hmm. It actually does a lot of things. It has functions. It detoxifies alcohols and drugs. It produces bile. It also produces various other things. Okay, but its function specifically in the digestive system is that it produces bile. So I don't want you to leave the class thinking that the liver only produces the bile. It does a lot of things. You can't live without your liver, and that's why it's called the liver, right? So it's one of the organs that probably has the most functions out of all the structures within your body. Okay, so I don't want to downplay its significance. But we say it produces bile because right now we're looking at just the digestive system. So it produces the bile. The bile is going to move into this duct right here. What is that duct called?
The bile duct.
Okay. And up this duct right here, which is called the
Pancreatic duct?
nope. The pancreatic duct is this one.
Cystic duct.
Cystic duct? And then when it joins together, it's called the, so this one actually is not the bile duct. Does anyone know what it's called? What's the term for liver?
Hepatic.
Hepatic duct. And then this one where they join together, that's the common bile duct. Okay, so the bile that's produced by the liver moves through the hepatic duct, up the cystic duct into the gallbladder. Why does it do that? Why does it have to go to the gallbladder? What's the function of the gallbladder?
It stores bile and concentrates it.
That's right. It stores and concentrates bile. So technically, can you live without your gallbladder?
Yes.
Yes.
Absolutely.
I don't have one.
And you are alive. There you go.
Yes.
You're evidence that you can live without your bladder.
That's how I know that you can live without it.
That's right. That's right. You're our living proof. So the reason for that is because it's not the gallbladder that produces the bile anyways. It concentrates it. It makes it stronger, but it doesn't produce it. The liver produces it. Okay. So people who have their gallbladder removed still produce bile, but it's just that when it's secreted, it's less concentrate. So sometimes that just means you have to avoid, extremely fatty foods. Okay. Because you're not getting it at the concentration that you need to, but you're still getting the bile. Okay. So the bile is produced by the liver, moves through the hepatic duct up the cystic duct to the gallbladder to get concentrated. And then whenever you eat a fatty meal, then that moves down the cystic duct, down the common bile duct, and then enter into the duodenum. Okay, so that's bile. So bile is one of the fluids, one of the chemicals that's being dumped into the duodenum, the small intestine. Okay. In addition to that, the pancreas also produces. The pancreas is super cool, by the way. The pancreas is actually another organ that's very difficult to survive without. And the reason for that is that it has both endocrine functions and exocrine functions. Okay, so it plays a huge role in your endocrine function. But here, because we're only looking at the digestive system, we're only focusing on the digestive functions. So the pancreas, in terms of the digestive function, it's going to secrete what? All of its pancreatic juices through the pancreatic duct and also dump into the duodenum. But let's identify, what does it produce? So bile is by the liver, but in the duodenum, you actually have tons of chemicals produced by the pancreas. Can you help me list them out?
There's a lot.
Yes, let's start with pancreatic amylase.
Then you have trypsin,
Hold on.
chymotrypsin.
You're absolutely right, but hold on. Pancreatic amylase. Pancreatic, it's produced in the pancreas. Amylase, meaning it's going to target what?
Carbs.
Carbs.
Because it's an enzyme. So pancreatic amylase is an enzyme that breaks down
Carbs.
Carbohydrates.
Yeah. Carbs. Okay. Then you have what? Pancreatic lipase, I heard.
Trypsin, chymotrypsin.
Carboxypeptidase.
Trypsinogen. Carboxypeptidase. Oh, carboxypeptidase. What else? So trypsinogen, procarboxypeptidase, one more of the peptides ones.
The chymotrypsin.
Trypsinogen. Good. Okay. And then a few more.
I have a question. Why would the trypsinogen and the chymotrypsinogen be like inactive at this point? Because it's
Beautiful.
produced in the pancreas, so it becomes active in the small intestine.
Excellent. That's
Thank you.
exactly it. Because all of these break down what?
Proteins?
Proteins?
That's right. Do you want the proteins in your pancreas to be broken down?
No.
No. And actually, that does happen in some cases because some of the fluids in the duodenum actually make its way into the pancreatic duct and into the pancreas and thus activates it. And then you have inflammation of the pancreas. And then that causes a lot of problems, right? And then that can lead to pancreatic cancer. And then that, you know, it's very problematic. So that's a great question. Why are all three in its inactive form in the pancreas? It's only activated once it gets into the duodenum, because once it gets into the duodenum, trypsinogen becomes trypsin, procarboxypeptidase becomes carboxypeptidase, chymotrypsin becomes chymotrypsin, because you don't want the proteins of the pancreas to be broken down. It's kind of cool how your body does that, right? It's like it plans everything out. Okay. So that, I mean, this isn't just random. I mean, this happens throughout your whole body. It's just beautiful how your body functions. Okay. All right. We're missing one more. What else is produced in the duodenum? So all of this is by the pancreas. This is by the
sodium
Sodium.
bicarbonate.
Perfect.
Yeah.
So why the sodium bicarbonate? Where did the food just come from prior to the duodenum?
It just came from
From
the
the
stomach.
stomach.
So it
And it's got
needs-
hydrochloric acid.
There you go.
That's right. So the stomach has a very particular lining that protects it from the acid, but only the stomach. So if that acid makes its way to the duodenum without being neutralized, it's going to start breaking down the small intestine. So you have to have sodium bicarbonate to neutralize the acidity or the hydrochloric acid of the stomach. Did you know that the hydrochloric acid in the stomach is strong enough to melt through metal? It can melt through metal. So that's why it's really important to have that nice mucosa lining along your stomach. Okay? But sometimes the lining gets broken, like either through Helicobacter pylori, which causes peptic ulcers, or by taking NSAIDs, non-steroidal anti-inflammatory drugs, that causes that mucus line to become thin. Okay? Anyways, cool stuff. Take human disease with me if you want to know more. Okay, so sodium bicarbonate helps to neutralize the acidity of the stomach. Okay, so that's why we say that the majority of chemical breakdown really occurs in the small intestine, which also makes sense then that what follows. So you've got chemical digestion in the small intestine, but you also have what?
Absorption.
Yes. Oops. Okay. So you've got digestion and you've got absorption happening here. Okay. So along the small intestine, what allows for the absorption is that the mucosa lining has absorptive cells. You've heard about this before. Where have you heard of absorptive cells before?
On the microvilli.
Yes. In what chapter?
That was in tissues, no?
It's, you haven't had urinary yet. Never mind. You have not heard about this before. Okay. You will hear about it in the next unit. So the absorptive cells also exist in the urinary system. Okay, so the absorptive cells have the microvilli because these two are connected together because they allow for absorption. Okay, so the absorptive cells contain the microvilli, which allows for what we call final digestion and absorption. Now the final digestion is through what we call contact digestion, because it comes into contact with it, breaks down into its basic units. So carbohydrates essentially would be broken down into the monosaccharides, proteins would be broken down to amino acids, and so on. And that's what is absorbed into the blood vessels. Okay, lipids are broken down to micelles, and micelles are broken down into the smaller functional units, and that's what is absorbed into the lacteals. Okay. All right. So absorption occurs mostly along the jejunum and the ileum. Okay. All right. Finally, we get to the large intestine. Someone tell me what happens in the large intestine.
It's the process of defecation, no?
Yep. But before that, what happens here?
It has to compact first.
And before that, what happens here?
The water absorption.
There you go. So there's still limited. There's still limited absorption happening here. There's limited absorption of water and some vitamins. And the reason why for some vitamins is because there's certain things that we cannot produce ourselves. But in the large intestine, what do you find that breaks down certain things to produce vitamins for us?
The microbes?
Yes. Okay, yep. So you have you have gut bacteria in your large intestine that breaks down cellulose, and that produces certain vitamins that we otherwise would not have been able to have in our bodies. So that's why there's still limited absorption of water and the vitamins, because the vitamins aren't released until the bacteria break them down. Okay? Then you're right, compaction. Oh, my mouse died, or my pen died. I tried to finish it all on time, but my pen died. So, I can't write it on there, but you can later on, because you'll get this PowerPoint. So make sure you add compaction and defecation. Okay?